You know, every single living thing, from a tiny bacterium all the way to us, relies on this one fundamental process to even exist, making a perfect copy of its genetic blueprint. This is DNA replication. It's this beautiful, unbelievably complex process that allows life to pass on its instructions. So, how does this molecular marvel actually happen? Let's dive in.
Okay, so here's our game plan for this explainer. We're going to start with the basic idea of how DNA copies itself, and then we'll look at the huge hurdles the cell has to overcome. After that, we'll meet the amazing molecular toolkit that gets the job done, unpack the story of the two different strands, tackle accuracy and the tricky end problem, and finally, see how all that new DNA gets neatly packaged up.
All right, first up, let's get right to the core concept. How does that famous double helix actually make a perfect copy of itself?
This is the big question, right? And what's so cool is that when Watson and Crick figured out the structure of the double helix back in '53, they pretty much realized the answer was staring them right in the face. The very shape of the molecule, with its two perfectly matched strands, just screamed out a simple, elegant way to copy it.
And here it is, semi-conservative replication. It's a beautifully simple idea. You just unzip the double helix, and each of those original or parental strands becomes a perfect template to build a brand new partner. The result? Two identical DNA molecules, but each one is a hybrid, half old, half new. And this wasn't just a good idea. The brilliant work of Messelson and Stahl proved it was exactly how it works. It's life's way of ensuring a faithful copy of the genetic code gets passed on.
But hold on, while the concept is simple, actually doing it is a whole other story. It's a staggering feat of biological engineering. I mean, trying to copy billions of letters of code with almost zero mistakes, well, that presents some massive challenges for the cell.
And this right here, this is the list of problems the cell has to solve. First, you've got to unwind this incredibly tightly twisted molecule without tying it into impossible knots. Then you have to keep the two strands from just snapping right back together. On top of that, the main enzymes that do the building, well, they can't start a new chain from scratch, and they're frustratingly stubborn. They only build in one direction, like a train on a one-way track. And through all of this, the process demands near-perfect accuracy. It's a tough gig.
So how in the world does the cell pull this off? It deploys a whole team of specialized enzymes. Think of it like a highly coordinated molecular construction crew, where every single protein has a very specific job to do.
First on the job site is helicase. Its job is to tackle that unwinding problem. You can think of it as the unzipper. It burns through cellular fuel, ATP, to power its way down the DNA, breaking the hydrogen bonds that hold the two strands together and prying them apart at a spot we call the replication fork.
But you know, when you unwind something that's twisted, the part ahead of it just gets wound tighter and tighter. It's called torsional stress, and it would quickly grind the whole process to a halt. That's where topoisomerase comes in. It's the stress relief expert. It works ahead of the fork, making these tiny, temporary cuts in the DNA, letting it swivel and relax, and then perfectly sealing it back up. Pretty clever, huh?
Okay, so the DNA is unwound and it's relaxed. But our main builder, DNA polymerase, has this really weird quirk. It's a master at its job, but it can't start from scratch. It can only add on to a chain that's already there. So to solve this, an enzyme called primase acts like the ignition switch. It lays down a short little strip of RNA called a primer. This primer gives DNA polymerase the chemical hook it needs to grab on and get started.
And with that primer in place, the star of the show finally arrives, DNA polymerase. It locks onto that primer and just takes off, reading the template strand and grabbing the matching DNA building blocks from the cell, adding them one by one to build the new complementary strand. This is the enzyme that's actually copying the genetic code at incredible speed.
Now we get to a really fascinating complication. Remember how we said DNA polymerase can only build in one direction? Well, the two strands of the DNA helix run in opposite directions. We say they're anti-parallel. And this creates a really clever but pretty lopsided situation at the replication fork.
Because of this one-way street rule, the two new strands have to be made in totally different ways. One strand, which we call the leading strand, is the easy one. It gets made in one long, continuous piece, just following right behind the helicase as it unzips the fork. But the other strand, the lagging strand, well, it's trickier. It needs to be built in the opposite direction that the fork is moving. So the only way to do it is to make it in a series of short, disconnected chunks.
These short little chunks actually have a name. They're called Okazaki fragments, named for Reiji Okazaki, the scientist who figured this all out.
Each of these fragments has to start with its very own RNA primer. Then DNA polymerase builds a short stretch of DNA until it bumps into the primer from the fragment before it. It's this kind of backstitching method, and it's the cell's ingenious solution for copying that lagging strand.
But obviously, you can't just leave a new DNA strand in pieces with bits of RNA stuck in it. That's just messy. So a cleanup crew comes in. A different kind of polymerase, DNA polymerase I, comes through and removes those RNA primers, filling in the gaps with the proper DNA. And then the final enzyme, DNA ligase, comes in and acts like molecular glue, creating the last chemical bond to stitch all those Okazaki fragments together into one single continuous strand.
So, the DNA has been copied. We are almost home. But we're not quite done. We still need to talk about two final, super important challenges. How to make sure the copy is perfect, and what to do about a weird little problem that happens at the very ends of our chromosomes.
To get that crazy level of accuracy, DNA polymerase literally has its own built-in spell checker. Or maybe a better way to think of it is a molecular backspace key. As it adds each new base, it double-checks its work. If it accidentally puts in the wrong one, it can pause, use its exonuclease function to clip out the mistake, and put the right one in before it moves on. This proofreading is what makes replication so incredibly precise. But there's a problem, and it's unique to organisms like us with straight linear chromosomes. Think about that lagging strand. When the very last RNA primer at the absolute tip of the chromosome gets removed, there's no way to fill in the gap. DNA polymerase needs a primer ahead of it to build off of, but at the very end, there's nowhere to put one. And the result? With every single round of replication, a tiny piece of the chromosome's end is lost forever. The cell's solution to this is just brilliant. Protective caps called telomeres. These are just long, repetitive strings of DNA at the ends of each chromosome that don't actually code for anything important. In humans, it's the sequence TTAGG, repeated thousands of times. These telomeres act as a disposable buffer. The cell can afford to lose a little bit of this junk DNA with each division, and it protects all the vital genes from being erased. But that leads to another question, right? If our chromosomes get shorter our whole lives, wouldn't we just pass on shorter and shorter ones to our kids? Well, to prevent that, our reproductive cells, the ones that make sperm and eggs, have a very special enzyme called telomerase. This amazing enzyme's only job is to rebuild and lengthen the telomeres, making sure that the next generation always starts with a full-length set of chromosomes. So how does this magic enzyme work? It's really cool. It's a type of enzyme called the reverse transcriptase, and it actually carries its own little instruction manual, a piece of RNA right inside it. It attaches to the end of the chromosome, and using its built-in template, it adds more of that repetitive telomere sequence onto the parental strand. This makes the strand longer, providing just enough space for the normal replication machinery to come in, lay down one last primer, and finish the job without losing anything important.
And finally, once we have two complete, accurate copies of the DNA molecule, there is one last step, at least for eukaryotes like us. We have to carefully package it all up.
You see, our DNA isn't just a loose strand floating around in the nucleus. It's incredibly long, so it has to be packed up super tightly. It does this by wrapping around these little protein spools called histones. This whole DNA protein bundle is called a nucleosome. Now, during replication, all of this has to be taken apart so the enzymes can get to the DNA. Then, right behind the replication fork, both of the new DNA molecules have to be immediately rewrapped and repackaged into new nucleosomes. So when you step back and you look at the whole thing, this microscopic dance, it's just incredible. It's not just some mechanical process. It's a high-speed, high-fidelity performance, choreographed with dozens of proteins all working in perfect harmony. And this dance, happening trillions of times in your body right now, is the absolute foundation of growth, of repair, and of the continuity of life itself. It is the microscopic engine that ensures the story of life is passed on, almost perfectly, from one generation to the very next.